This week on the Galactosemia Podcast, I talked to Clayton and Mandy from BridgeBio. BridgeBio is a pharma company that's working on gene therapy for galactosemia. And honestly, when I say the words gene therapy for galactosemia, a whole mix of feelings rush in, right? There's hope for what could possibly be a cure. There's nervousness for just not really knowing a lot about gene therapy. There's the sense that we're talking about something that might have once felt like it was science fiction, but is now very much a reality. So in this episode, we break down the basics of what gene therapy means, what the different kinds are, where BridgeBio is in the process, and we have some real personal conversations about what it might mean for families and the considerations. Personally, I know there's a lot of considerations that my wife and I are going to have when it comes to something like gene therapy for our son. So I asked them about their approach and how parents can weigh those risks of hopes in something that's as big as gene therapy. This is easily one of my favorite episodes so far, and I really appreciate Clayton and Mandy for being so open. You know, in the pharma space, it's not always easy to have these conversations. There's a lot of red tape and there's a lot of legalese, but they've been very kind and open and willing to have these conversations, so it leaves me hopeful, and I hope you enjoy this conversation. As always, these episodes are available early and ad-free on the Galactosemiapodcast.com. You can sign up there if you'd like to support the show and help to keep this going. Thank you to all of our supporters who are part of the site. Okay, without further ado, here's our gene therapy episode with BridgeBio.
The Galactosemia Podcast.
Okay, I am very excited to talk to my guests today. You know, if you've been listening to this show, you've probably heard me talk about gene therapy in some fashion or another. It's something that I have been really interested in since I heard that there was someone or some companies working on gene therapy specifically for galactosemia. And I'm really happy to welcome two people from this show that I consider on the front lines of that effort. My guests today are from BridgeBio. It's a company that is actively working on gene therapy for galactosemia. Clayton Beard is the Chief Scientific Officer at BridgeBio, and Mandy.Rohrig is the Senior Director of Patient Advocacy at Bridge. Mandy and Clayton, welcome to the show.
Thank you. Great to be here.
Hi, Ryan. Good to see you, and thank you.
It's so good to have you guys on. I want to do a little bit of table setting for this episode before we dive in, because I think context is important. Mandy, you and I connected about 6 months ago. Britt Cudzillo gave me your information, and I cold emailed you just asking to talk. And since then, we've got to know each other quite a bit. And in June, there was even an event that was held in Baltimore where members of BridgeBio gathered at Catalent, which is a leading manufacturer in drug development. We did sort of a patient day where we got to share some of the patient stories of those living with galactosemia or raising kids with galactosemia. We learned what BridgeBio has been working on. We learned how Catalent kind of factors into that whole picture. It was really an educational experience for me. And so what I'm hoping today that we can do is dive into some of that nitty-gritty of what the gene therapy means, what you guys are working on, and really what the hopes are for our community. And we can kind of all base it in reality and in science and in what we know right now, because I think it's so important and it's so hopeful for a lot of members of our community. So with that table set, I would love to start with just a little bit of background from each of you so people can get to know you. Mandy, why don't we start with you?
Sure. So I'm going to can I add to your table setting?
Absolutely.
So
Please do.
The learning experience went both ways. I think the individuals who joined from our company and the leadership and individuals from Catalent also found the experience to be incredibly informative, enjoyable, and it's something that they hope to do again in the future. So thank you for that. My background is as a physical therapist. I worked in the clinic with people who have neurological conditions, specifically multiple sclerosis, Parkinson's disease, balance and vestibular disorders for over 15 years. And during a little time called COVID, you might both remember that, it became incredibly difficult to touch people. So I transitioned out of physical therapy and began work in industry. And all along the way, I've worked in nonprofits. One in particular, Can Do Multiple Sclerosis. It's a multidisciplinary wellness program for people with MS and their families. And I share that because it's relevant in the fact that part of how I think about education and part of how I think about working with people in communities is very holistic, because I think the multidisciplinary team with the family and the patients and the community at the center is really how I think and act in this job and certainly in my previous work as well.
And now you find yourself kind of the point person for patient advocacy at Bridge, and you have a lot of touch points with the communities and the people in the rare disease. Yep.
I've been serving as kind of that liaison between the company, people living with the different conditions, the communities, and the nonprofit organizations for almost 5 years now. Yep.
And Clayton, what about you?
So I am a virologist by training. So I've always, I did my graduate work in understanding the genetics of viruses, and my initial work was making vaccines, so ways to prevent viruses from infecting us. And along the way, I started to realize the beauty and the utility of using viruses to actually do good things for health. So that was using viruses to make vaccines against other viruses. And then along the way, gene therapy started to become a concept. And I watched that go from a concept to a reality about 15 years ago when they really started to dig in. And so I began working more with using viruses for gene therapy. And over the past few years, have really started to be involved more with the communities of people who we're trying to make therapies for. And that's in great thanks to Mandy and her colleague David and BridgeBio. But it's been extremely rewarding, and I've really become much more motivated, and it's changed the way I think about science and the way I think about developing medicines for the communities, and really listening and getting to know the people that we're trying to help, and understanding what they need and how best can we meet those needs. And again, of course, I put in my bent of how can I use our friend the virus to meet those needs. So, yeah.
And I would say, after talking with you probably in total for a few hours along like how long I've known you, like we've had some really like lengthy conversations, I would say I put you in this tier of people that I consider like the smartest people that I've ever met. It's like Dr. Barry's in there. You're definitely in there. There's like people in there that I have to kind of slow down and be like, okay, I'm going to ask you a couple stupid questions that you can like rewind and say again in a way that I understand it. I want people to know that, like, at least from my experience talking to you has been, you are so smart and you just know what you're talking about in a way that like makes me feel confident that I can believe what you're saying.
No. Well, thank you.
You're welcome.
We do try to put a lot of effort into scientists in general, we kind of speak our own language. And so I do like to think that we have a little Berlitz translation book on like how to make it so that it makes sense. And I would hope that the chief scientific officer of BridgeBio would be a very, very smart man or woman. So like right. It's a good thing. Yeah. So I do want to jump into gene therapy, gene editing, gene replacement, this topic, especially as it relates to galactosemia. But I think if we can first start with how did BridgeBio get started? And I think I read somewhere that the CEO, Neil, had an idea for a different business model than traditional pharma companies. Can you guys shed some light just into like how Bridge approaches things and how that might be different than other pharma companies? Sure.
Okay, you want to start, Clayton? Go ahead, and I'll build.
I'll start, and then Mandy can correct me. So Neil Kumar, our CEO, actually had taken a class from a professor, and that professor had written a paper, and that professor's name was Andrew Lowe, and Andrew proposed this novel business model, and that was one of having a decentralized company that really focused on multiple different disease areas or specific therapies. And the whole idea was, how many therapies would you have to work on until you get to a point where you're guaranteed that one of them is going to be successful? Because I think that's the real hard part about drug discovery, is most of them fail. They're very difficult to get a drug successfully developed. So this model was one of a single entity that was subdivided into 18 or so different small affiliates. And each one of those was focused on developing a therapy for a particular rare genetic disorder in the case of Bridge. So that was sort of the novel model that Andrew had proposed, and Neil decided, hey, let's give that a shot.
Mm-hmm. And how does galactosemia, where did that fall in? Was it part of one of the affiliates? Was it something from the beginning? Like, where did galactosemia fit into that structure?
Yeah, it is one of the affiliates. It is because each affiliate is focused on one genetic disorder to look at. And so we have a couple of gene therapy affiliates. So they are grouped into sort of a sub-affiliate called BridgeBio Gene Therapy, and that's where I'm part of the leadership of that sub-affiliate or larger affiliate that has the gene therapies in it. But galactosemia is just one of those targets that Bridge is focused on.
And how or why is it beneficial to do affiliates? And like why does that help rather than just what, I don't even know how traditional pharma has worked, but like why is it beneficial to do it this way?
Well, I think one of the big benefits is you're able to have smaller teams. So because all of those affiliates are sharing a lot of the infrastructure of a company. So when a company gets to be, of a good size, the infrastructure that it takes to run payroll and to administer benefits, but then also a lot of drug development has shared resources around regulatory interactions and legal teams and clinical teams. So each affiliate is able to go after and start working on these therapies for a much less amount of money, for a lower spend. And so we're able to go developing more therapies for the same amount of money because we're spending less on each individual one. Is that accurate,
I was just
Mandy?
going to say, Clayton, could I ask a question to just clarify? I think it's efficiency and the model allows for efficiency and resource management, but also time, because you have dedicated teams with expertise in those particular conditions. Is that correct as well? Yeah.
Right.
Yeah.
So the company as a whole has some really talented generalists, generalists, great regulatory people, great legal people, but then each affiliate can really go out and hire the best galactosemia scientists that they can find.
Right.
Mm-hmm. And that's still a very efficient way to go because that affiliate's not having to hire the legal and all the other shared resources. Yeah.
And you mentioned that, there's other diseases that you guys have worked on or still working on. I'm curious, have you guys developed therapies that have been approved that are out to market right now that are treating other diseases?
Yes, we have. We have our most recent one is a product for a cardiac condition called ATTR. And that's part of the model as well. ATTR is a rare condition, but it's not near as rare as many of the ones we work on. Mm-hmm. So that commercial drug will actually bring in revenue that we can then use to fund research and treatment in these really rare conditions.
Right.
Because the problem with a rare condition is you're never going to sell enough product to make your money back.
Yeah, I mean, galactosemia is kind of like a model for that. Like, let's say you guys come up with a gene therapy, like you can sell 3,000 of them in the U.S. and then you've like cured everybody of galactosemia. So it's like that you're capped at a rare disease.
Right. But it's one of these, because we have several products that have much larger populations, the revenue from that is funding and enabling us to work on these products that no single company could ever sustain itself on working on just galactosemia, for example, or any other small. So that's a big advantage to this model, where you have all of these affiliates. Some of them bring in enough revenue to allow other affiliates to pursue therapies that otherwise wouldn't be sustainable.
Okay, great. So that's a little bit of background on BridgeBio. Let's talk gene therapy. Let's get into it. I know just from talking with you guys that you guys are working on gene replacement therapy, and that's different than gene editing therapy. Clayton, can you give us let's say like a 7th grade biology, maybe 8th grade biology lesson on gene therapy? And like, I'll stop you if I'm in like a 4th grade, which might happen, but let's start there. And can you kind of give us the 101 of what this all even is?
Sure. And whenever I talk about gene therapy, I like starting to make sure we're on the same page. What do we mean when we talk about a gene? And for the context of this conversation, a gene is really just a sheet of instructions that tell your body how to make something it needs. Okay. And when we talk about a genome, that's a collection of all those instructions. So that's like a book with all these pages, and each page is a gene. Each gene is instructions that tell your body how to make something it needs. So
we've got a lot of other genes that don't even have to do with like GALT enzyme, right? We've got genes that
You've
are
got
just like
thousands of them, but you have one gene for your GALT enzyme, and that gene, when I say a gene, it's just the instructions your body uses to make GALT enzyme. And when somebody has galactosemia, something's happened to that page of instructions that when the body tries to read it to make GALT enzyme, it can't make an enzyme that works. And without that enzyme, you have galactosemia. You can't metabolize galactose. So in gene replacement therapy, we're using, again, our friend the virus, to bring in a new page of instructions to the cells in your body. So that's all we're doing, is we're putting in a page that the cells of your body now can read those instructions and now make the GALT enzyme.
Okay.
And that's gene replacement. That gene that's no longer legible and no longer the right instructions, we're putting in a new gene.
And that is different than gene editing because, just a guess, gene editing would not be adding in a new page. Is it like erasing the instructions?
Exactly.
And
it's
instructions?
Correct. Exactly right. Gene editing is going in and correcting the mistake in that original page of instructions.
In your virology, what was your
virology?
You're and you were a virologist. I think of it like meteorologist, but it's just viruses. It's like virologist. But in your under your virology hat, when you see the difference between gene editing and gene replacement, I imagine it's not that one's better than the other; they're just different. But what are maybe pros and cons when it comes to each?
They're both great technologies. There are some genetic problems that gene replacement is going to be better for, and there's some that gene editing is going to be more suited for. We're doing gene replacement right now because gene replacement has been in development much longer. So gene replacement started easily 15 years ago, if not 20, 25 years ago. It's been coming along, and all of these new therapies take a long while until they're ready to be used in humans and widespread use. So gene editing is a great technology. It's just not as advanced yet as gene replacement is.
I've heard a lot about CRISPR. What's CRISPR?
So CRISPR is a tool. That's like saying that's the tool that you can use to go in and erase and rewrite part of that sheet of instructions.
So that's editing, gene editing.
That's gene editing. So CRISPR is gene editing. Yep, exactly.
Got it. And there was actually in the New York Times recently a really good story about gene editing that was quickly approved through CRISPR to save one boy's life. But we're talking about gene replacement for galactosemia, and that's a little bit different. You once told me about an analogy that I wanted you to give of like envelopes and things.
Sure.
Can you remind me of what that was and where that fits into all of this?
Yeah, that's great. I'm happy you remember that. So when I talk about putting in that new page of instructions and that we use a virus for it, the analogy I use is that new page of instructions is like a letter that you would fold up and you put it in the envelope. And then the envelope is going to deliver it where it needs to go. And so depending on where we want, what part of the body we need that new page of instructions to go to, we use a different virus. And when I say virus, it's just the outside of the virus, and it really acts as an envelope. So we use that to take that new sheet of instructions into the body and to the different parts of the body where it needs to get to. So for galactosemia, we're using an envelope that delivers to lots of different parts of your body, to your brain, to your liver, to your muscles, because all of the tissues of your body, it's important that they are able to metabolize the galactose that's being made inside them and also being taken in by in your diet. Whereas other gene replacement therapies, things like there's gene replacement therapy for hemophilia, that one uses an envelope that primarily puts that therapy into the liver because the liver is where those blood factors are made.
So, Clayton, different gene therapies have different envelopes or viruses that are used because you want to target different tissues?
Exactly right.
Okay.
Yep.
And so one of the things that actually Dr. Judy on this podcast a couple weeks ago talked about was that we have a pretty good idea. In fact, she did a study, which we talked about on the episode, of bone marrow transplant, where they had a theory that if they did bone marrow transplant, they might be able to get galt activity restored. And they were able to get it in some places, but they weren't able to get it restored in the cells that she thinks really has a high likelihood of mattering a lot, which is the liver and the brain. And so if you're targeting different areas, I assume the envelope or the vector that you guys are using can target those brain and liver areas?
Yes, exactly right. The envelope that we're using does target the liver and the brain.
From a science perspective, how do you even get to a place where you know that? Like, how can you tell that?
Yeah, that's a great question, but that's where academic research and just research in general pays off. There are labs, there are people who have devoted their careers to isolating different viruses. And so the virus that we use is called AAV, and that stands for adenovirus associated virus. so adeno-associated virus. And it's a little virus that is always associated with a big virus called an adenovirus, hence the name.
Okay.
But AAV doesn't cause any disease in humans. It's just sort of a hitchhiker that tags along with this other virus and needs it in order to be able to survive. But there are hundreds of different AAVs, and they're separated by where they like to go, what tissues they go to in somebody's body. So over the years, many years, lots of research, people have studied those viruses and determined what tissues they go to best.
And we found that the one that you guys are using for gene therapy for galactosemia, is it just the research has shown that it does a good job going to the brain, that it targets the brain? Like, how does it, is there something you can measure to say like, oh yeah, that is activating in the cells in the brain?
Yeah, so it's a good question. So your brain is very good at keeping viruses out of it.
Yeah.
Your body is sort of engineered so viruses generally don't get to your brain.
It's what we hear about, like the blood-brain
barrier.
Right?
Exactly.
Yeah.
And the AAV that we're using, the AAV that we took apart and used as the envelope, is a very unique AAV in that it can cross the blood-brain barrier. So it's been studied in, mice originally and in monkeys, and then it's been used in a lot of humans. And so we do know that it does cross that barrier, and it does deliver those new instructions into the cells of the brain, into the CNS, the central nervous system.
And is that the same for the liver too? You just kind of know that it goes to the liver?
It goes to the liver the same way. And a lot of viruses go to the liver because that's your liver's job, is to filter out things. So many of them go to the liver. Almost all of them go to the liver. But they also, different subtypes of AAV go to different tissues in addition to going to the liver.
Do you know if, whether it's developed by you or not, or other people, I don't know, but if this vector, this envelope that we assume targets the brain has been used for other disease therapies?
Yes, it has. We've used it for a condition called Canavan disease.
Okay.
And that is a severe neurological genetic disorder.
Okay.
That we have developed a very similar therapy for, a gene replacement for Canavan, and that instead of GALT in the case of Canavan, it's a gene called ASPA.
Okay.
So we use this exact same vector, this same envelope, to deliver instructions on how to make ASPA into those patients.
And that, I don't want to spend too long on that drug, but did it go to clinical trials? Did it come out of clinical trials?
It's still in a clinical trial right now.
Okay.
Yep.
Okay, and so that's essentially how we're trying to get gene therapy with galactosemia. Is you are putting these new instructions in an envelope or a vector that is going to target cells in specific areas of your body, the brain, the liver, I imagine the muscles, and these cells will hopefully turn on, with these new instructions, the ability to have that GALT enzyme process galactose.
Correct.
What have you guys tested so far in terms of this work?
So far we've tested it in mice. So there have been mice that have been, the instructions for the GALT gene in the mice have been altered so that those mice cannot make GALT. And so those mice actually have mouse galactosemia.
We actually talked to Dr. Judy extensively in her cohort, who they have rat models, but I know Dr. Lai has mouse models, and so we've talked to them about, or we talked to Dr. Judy about what is involved in making a rat model have a knockout GALT gene. And so you're talking about that in mice, of testing that knockout GALT gene in mice and giving them this gene therapy?
Exactly. So you mentioned Dr. Kent Lai. He's who we collaborated with.
Okay.
So Dr. Lai has a mouse strain that has the instructions for the GALT gene have been altered, so those mice can no longer make their GALT protein. So we're able to, in collaboration with Kent, we're able to treat those mice with this envelope and containing the new instructions, and it delivers to those the cells in the mouse, and those mice are able to make GALT and are able to start metabolizing galactose.
And did it end after mice, or were there other animals, and did you study it in other animals as well?
So for those kind of studies, we've only done in mice.
Okay.
We're currently doing safety studies in mice and in non-human primates.
Safety studies in mice and non-human primates, so like monkeys.
Yes.
Yeah, and that's pretty typical, Ryan, correct, Clayton, where research in the preclinical stage, so before it goes into people, that the scientists and the research will evaluate the potential therapy in mice, in cells, in non-human primates or monkeys, right? That's a pretty typical process of development.
Exactly.
Mm-hmm.
Yep.
And what did you see in the mice whenever you guys studied this gene therapy?
So these mice have some similarities to what a person with galactosemia would see. So in these mice, they have an increase in a chemical in their blood called galactitol. And they also have increased levels of something called Gal-1P. And both of those are molecules that the GALT enzyme ultimately leads to their being processed and going away. So when these mice don't have the GALT enzyme, the mice have an elevated level of Gal-1P. And for those of you who either have a child with galactosemia or have galactosemia, you know that is something that the physician will look at. They'll take blood and they'll talk about the Gal-1P levels or the galactitol levels. So those are elevated when you have galactosemia, and they're elevated in these mice. And then after we treat the mice, the levels of Gal-1P and the levels of galactitol return back to the same level that you would see in a mouse that did not have galactosemia.
That's so interesting. How did you guys test the blood to get those levels of galactitol and Gal-1P?
Yes, we've tested blood, but because they're mice and they're laboratory animals, we also looked in their brains and in their livers and in their muscles to make sure that we're seeing those levels go down in all of those critical areas. Again, to get to your question before of how do you know that the envelope is getting the new instructions to those tissues, that's how we know. We look in the mice to see that that's where it's going.
Okay. Were there any, in that study, when you guys did that with the mice, were there any off-target effects that you noticed in the studies so far? Like by off-target, for those listening, we mean like any unintended impacts outside of what the therapeutic target is?
None that we've been able to see. We've tested this in toxicology studies where you specifically look for any adverse event, any changes in blood work or tissue or anything like that, and it's been fine. There's been nothing that we've seen that was indicated any kind of toxicity associated with it.
Interesting. And so you guys studied that in mice. It seems to have gone at least well enough that we're still talking about it, and we're on this call right now. At some point, this will need to be tested in humans if it is deemed successful. What does that process look like, getting from mice to humans?
Yeah, so that process starts with the work that I was talking about, and all of that is is preclinical work or non-clinical work, and that's putting together a data package, and that's what we're doing with galactosemia right now. But that's accumulating a set of data that we then combine into a filing that will be shared with the FDA and what that data package is going to do is it needs to demonstrate that we understand what the problem is in galactosemia, explain how this therapy is going to help correct that problem, and then demonstrate that we understand how that works and things like what kind of doses are going to work best and what age of dosing is going to work best, and the overriding data that we have to have is that of safety. So that's the FDA's number one concern, that the therapy is safe and that it's going to be effective, that it's going to give some reasonable expectation of benefit to the overall health of somebody who gets it. So the data is all centered around those two things. So safety data and then demonstrating that it's going to be beneficial to the overall health of the person who gets it.
And so you guys put together a package that gets submitted to the FDA, and the hope is they say, yes, like we agree with your assumptions and your data and everything that you've put together, and we're going to allow you to go into a clinical trial where, from my research, it sounds like clinical trials are more studies of what you were just talking about, because you have different phases. You have a phase 1 where it's really just studying, is it safe in humans? You've got another phase where does it work and at what dose does it work? And then phase 3 is more large scale efficacy and safety, which is a lot of what you were just talking about and what the FDA is considering in your initial package as well.
Correct. So that's interesting. I've never looked at it that way, but in the preclinical work, we kind of go through all 3 of those phases, and then when it goes to the clinic, it starts over again.
Yeah, and you have to kind of prove it.
It's a really good analogy. I hadn't thought of it like that.
That is good. I haven't. Yeah.
I'm just thinking, Clayton. So it takes a long, long time to get that preclinical package just right. Like, it's years of refinement, it's years of finding the right dose, it's years of analysis. Can you talk about, because I genuinely don't know, like, how long does that generally take to get through that preclinical stage till you are at a point where you're like, hey, we have something, here's a nice package, let's go talk to the FDA?
Yeah, that's a great question. It does take a long time. A lot of it depends on how much work has been done on the condition ahead of time. Mm-hmm. So for galactosemia, people have been studying galactosemia for decades. So that really helped us step in and be able to understand what's happening in galactosemia and what approach might be therapeutic and would help them. Dr. Lai had been working his whole career on galactosemia, mm-hmm. so we were able to partner with him. So there's a lot of things that will affect the overall timelines, but in general, it's been my experience that it usually takes at least 4 or 5 years to get that preclinical data packaged together. Now, that can get to be shorter the more therapies you work on, because your team gets a little more expertise in it. But then also the FDA really wants to help us move these therapies forward. So they will allow you to reference similar work. So it kind of lowers the volume of work that you have to get done.
So it sounds like the answer to Mandy's question is kind of like it varies and depends on a lot of different variables, one of them being like how much work has been done in this area already.
Yeah.
Right.
I was just going to add, so just for the benefit of the broader community, it may feel like when we talk about that we are, for this program, we're still in the preclinical stage of development, it may feel like forever. And it's because of all of these characteristics that Clayton just outlined, that it does take a long time to get it just right. We don't want to be on the dartboard. We want to be on the bullseye if we're going to take it to people.
Right. And how long have you guys been working on galactosemia specifically? You've been doing preclinical and all the research and the studies for how long now?
So the preclinical work began about 4 years ago.
Okay. And so at 4 years now, like you said, you're at the point where you guys feel confident enough to start putting together this packet of information.
Yes.
And I just want to clarify too, Ryan, like we're still working. There's still work to be done in this preclinical phase. So we're still pulling together the elements of that package to make sure that package is just right and the way it should be.
Yeah.
Yeah.
Yeah. And we know the community, I think, of galactosemia know from other experiences that we've had with the FDA. I won't go any further than that. Know that it can be very hard to get a drug or a therapy or whatever approved, even after members of our own community have been part of trials. And go listen to some other episodes if you want some more detail on that. But it with the FDA, you do want to get it. You want to put your best foot forward, and it seems like you kind of have one shot of like, hey, this is what we want to do with a clinical trial. There's really no telling how long it's going to take them to consider everything. There's no telling like what exactly they're going to be kind of digesting with all of this. So you're just trying to make your best case, it sounds like.
Yep.
Obviously the hope is that this does get approved by the FDA. That's everybody's hope. I think that obviously the hope is that it's safe and that it's effective and that it is approved, and then we move on to clinical trials. And if we do that, it struck me as I was prepping for this that people who listen to this podcast, specifically listeners of this podcast, are theoretically people that might participate in a trial like this. Or have a son or a daughter who might participate. My son, Elliot, is 1 year old. He would probably be a kind of like a prime candidate for his classic galactosemia. He has no enzyme activity. So I, as the parent, that's something that's a very, very huge decision that I then need to consider of whether or not I even look into thinking about should he be part of a clinical trial like this, if he's eligible. So I want to kind of focus a little bit on that and see if we can get some idea of what that participant experience might be if we lived in this world where we were doing clinical trials. Do you guys have an idea of what preferred age group you would want to test it on?
I mean, I don't think we really have specific clinical trial structure or clarity at this time.
Sure. Okay. That's fair.
Yeah.
That's fair. And I know we have to make sure that we don't want to set expectations for something that might change, especially when it's all going through the FDA.
Yeah, thanks for saying that, Ryan, because I think that's exactly it. We don't know with certainty, so until we do, we want to make sure that we're being as thorough and doing our diligence before we share that broadly.
Are those things like the experience of the patient, what age group you guys are targeting, how often somebody is going to have to go somewhere, or will they have to have a lot of blood drawn? Are all of those questions something that is considered by the FDA while they're considering the application? Or is that something that, like, if it gets approved by the FDA, then we tackle all of these questions?
Ryan, this is why we at BridgeBio and many companies engage with communities early on, so that we can start having these conversations in advance of creating a clinical trial design and in creating a clinical trial framework. We want to make sure that how we design the trial, how we think about the trial, how we think about the outcomes that we want to measure, that we've had those conversations well in advance of ever submitting a package to the FDA, that we've thought through that. So we know that a community is aligned with how we're approaching and thinking and measuring and taking action in this particular trial. And I don't know, Clayton, if you want to add to that.
Yeah, I think it's important too that in that IND application, we write a clinical protocol. So all of that you're talking about, we have to think through and plan out, and we put that in that proposal, and the FDA gives us their thoughts back on that. So they're very much part of the process of determining how that trial is run, and things like the age you start in and how often the blood draws are, and what kind of testing you do to see that things are safe and things are working. And so they're very, very involved in all that with us.
Yeah, as they should be. I mean, it's such a big thing, right? A clinical trial, and especially if you have young ones going through it, like, you know, we've talked to families on here whose kids have been through clinical trials and how much goes into all of that, the blood draws, the multiple days it was in Atlanta, like all of the travel, all of everything. It's a lot. And so it should be considered carefully. And so is that something you mentioned that you are still in the mode of, like, trying to pull together some of this data for this application? Is that one of the areas that you're still focused on, like, coming up with is what the best, are you looking at the community to kind of help inform that what trials can look like?
Yeah, absolutely. I mean, we are, which is why we appreciate how, you know, our engagement and our relationship with you. We appreciate our engagement and relationship with the Galactosemia Foundation and the broader community, because these questions are critical, and how we shape this trial is really guided by the information that you all provide us. So yes, absolutely. Those specifics are things that we're talking about with the community, and hoping to continue to get more information.
Is there a want to reach out to more members of the community? Like, if someone's listening to this, is what I'm getting at. If someone's listening to this and they're like, man, I really want to talk to Mandy about whatever they got going on over there.
We'd be cheering them on.
Are you willing to, like, receive emails from people in the community?
Absolutely. Absolutely. We talk to people in the community who reach out to us all the time, and we're delighted to talk to them. And then if it's at an odd hour of day or night, I just give it to Clayton, because I don't answer emails at 2 a.m. But I don't think Clayton ever sleeps. No, truly though, we are here to listen.
Okay.
And we are here to help people understand and have those conversations. Very similar to you, Ryan.
Yeah. Well, if it's okay with you, I'll put your email in the or an email that people can reach out to. Might be a catch-all email or whatever. But I think that's important, because I do think there's a bunch of people in the community that are probably curious about this, that don't know how to get to that entry, like get into, like, who am I talking to? What are we talking about? I just want information. But I know through our conversations that, like, it's actually quite easy to talk to you guys about it and to get your real thoughts, because it is so important. It's important to you guys, and you obviously know it's important to us. So it's like we're all kind of working towards the same goal and the same focus. And I would imagine you guys are gonna be at the Galactosemia Foundation conference that they hold every other year.
Yep, next year it's in Aurora, Colorado, I believe.
Yeah, Colorado.
Sure.
So we'll be there. You'll be there. If nothing else, people can run into you there and maybe even listen to you give a little talk or a presentation. I'm not sure. I don't know if you've set that up, but
we don't know
at
either.
least shake a hand. We don't know either. We're all wondering. In fact, I would love to do a live Galactosemia podcast at the Foundation's conference, but I think I emailed somebody on the board and was like, "Hey, can I do this?" And I haven't heard back. So none of us know what's going to happen yet, which is okay. We'll figure it out.
For sure.
I have a question that I'm just going to ask because it's something that I think about a lot, and it's a little bit serious, but and I don't know that you'll have a great answer for it, but I'm a dad, right? I've got a 1-year-old. He's got galactosemia. It's not something like some of these other rare diseases where it's a life or death scenario, like this one we were talking about that they wrote up in the New York Times, where if this kid doesn't get this gene therapy, he most likely passes away. Elliot has galactosemia, but he's meeting a lot of his milestones right now. And we know just from talking to so many different people in the community that there is such a wide range of how galactosemia shows itself in different people. Some people have severe effects. Some people have really no effects except for they need to change their diet. I don't know where Elliot sits in that. I do know that gene therapy is a really, really, really big consideration of like, that's a huge step. Like that's a big thing. And the risk reward of a gene therapy for this rare disease, when you don't know what the other side of that looks like if he just lived his entire life with galactosemia, is something that I'm like grappling with. It's something that my wife and I talk about often. It's something that I think about a lot because you don't want to make the wrong choice. You don't want to choose gene therapy and find some side effect that you just regret, or you don't want to not choose gene therapy and maybe a bunch of other people chose it and they're cured and your son or daughter is having a really hard time with galactosemia. And all of those things are in play because we just don't know. And so I guess my question is like, how do you guys approach knowing that that's something that a lot of the community is going to have to grapple with if this moves further and further and further as we hope it does and we expect it to? Like, how do you guys approach that?
Yeah, I think you're seeing that today, that we're here, we're talking with you on this podcast because our goal is to help the community understand what this therapy that we're developing, what it is, and enable them to just understand the potential and understand the risks, and to also make them comfortable with, like you said, reaching out, having conversations. Mm-hmm. Ultimately, we're extremely aware and sympathetic that it's a choice that every parent or every person has to make for themselves. Our part in that decision is to make sure that you have clear, accurate information from us on, at least for our part of it.
Mm-hmm.
Yeah. And that's great. Like that's what I think that's all that you can do in you guys' position.
Right.
And when it comes down to us as parents, like there's not much more we can ask for from you, because I wouldn't want guidance of like you should or shouldn't do this. I just want the facts of what it looks like and what the data says. And then like obviously we have to make a decision. Mm-hmm. It's just, and you are both parents, so I know you guys, both understand what it's like to have little ones. It's your whole world, mm-hmm. It's everything. And gene therapy is something that 30, 40 years ago, it wasn't even really a consideration. And now it's a reality for a bunch of diseases, and could be a reality for galactosemia. And so, it's getting to that point where I think a lot of the parents in our community are going to have to make those considerations.
And I think if you can help guide us, Ryan, you and other parents in the community, to the information that helps inform your decision, that's something we can also, partner with and do together and create educational materials or guide you to the American Society of Gene and Cell Therapy that has a lot of great educational materials so that you can engage in those conversations with trusted people like your healthcare providers or other family members, because we just want to arm you with that information that's helpful.
I love to hear you say that, because I'll tell you exactly the types of things that I think would make. I don't know if it would get me there, but I know it would definitely be something that helps kind of rest some of my worries. Is I would really love a look at the data of what has been studied, and I don't work in pharma. I don't know if that's something that is traditionally shared, not shared. I have no idea, but I know that as a parent, it's going to take a lot for me to cross that line of yep, let's do it. Yeah. And I am somebody who people on this podcast know I'm not feeding Elliot the hard cheeses that people say that we've studied so far that say we can give them sharp cheddar, because I haven't studied it myself, because I haven't tested the levels of sharp cheddar and the galactose in that. And that's something that I am going to do at some point. And maybe that will make me feel better. I will see it for myself. I know how the tests were ran. I know the data behind it. And it would be for even more important for something like gene therapy. That's why I want to really understand it. I want to know, off-target effects, doses, how many mice, and then once we get into the clinical trials, it's going to be important when we talk about dosage and age of kids who had it, and all of that stuff is it's going to be so important, I think, for the community to know, or at least it would be important for me to know, to feel good about putting him in a trial. Yeah.
Well, Elliot's lucky to have you as a parent. It sounds like someone who's,
I don't know. He might
disagree with you when he's a teenager.
Yeah. He's like, Dad, lay off for a second.
As the parent of a teenager, I suspect there will be a period of time where it will be the layoff dad.
I think there will be, and I'll say, listen, I brought you into this world. Yeah.
I think what you said.
Your mother and I can take you.
I think what you said is very important, though. Again, we aren't here, as Clayton mentioned, to try to convince people or not. Our role is to really provide you with reliable information. Information that you need to make decisions on behalf of your child, or on behalf of, and for those of your listeners, on behalf of themselves. And that's information we often like to create with the community, because you all are the true experts. You live with it every day. Clayton's smart, but I don't know, Ryan.
He is. He's very smart. He doesn't have my podcasting skills, but he's very smart. No. Is the mouse data something you guys have available? You would consider making available?
Sure. I mean, we will publish that mouse data. You mentioned
it will be published?
Yes. And okay. You mentioned the Galactosemia Foundation meeting. That's the kind of thing that we would present there.
Okay.
Because that's one thing I really enjoy about BridgeBio is they're very transparent and open, and are big believers in sharing all of that information. So that view that I shared of not forcing a decision but just wanting to educate, that's not just me. That's right. Sort of the value of the company, and I think that's why Mandy and I and so many others are still part of it.
Yeah. And I can add too, Ryan, we have co-created with communities that we've worked with in the past webinars. I don't know if webinars still have any popularity at all, but we try to meet people where they are with the information that they might find helpful in their decision-making process. So that can be webinars, that can be podcasts, that can be paper handouts.
Right. I think that goes a long way, and that's that effort is it's what I believe is effective as well, because obviously, like, I have this podcast, but whenever Elliot was diagnosed with galactosemia, the information was a little too hard to get for me, and I wanted somebody to meet me where I was at, which is in the podcast world, right? Mm-hmm. And so this type of thing goes a long way. I think, like, is there a place where people can go to learn, like, let's say people want to know what other drugs BridgeBio has developed and has gotten approved by the FDA, or they want to do a little more research. Like, is that information readily available anywhere?
About the business, you can find on bridgebio.com. That's our company's website. If people reach out to me about information to learn more about galactosemia, I encourage people to get in touch with the Galactosemia Foundation. If people are more interested in learning about gene therapy, the American Society of Gene and Cell Therapy has an entire educational webpage and a number of videos that are really well done and helpful to explain some of the things that Clayton touched on today. I think those are some of the reasons. And I would probably give them, if you're okay with that too, Ryan, I'll add your podcast to the list. I haven't asked your permission for that. Now I'm doing that publicly.
For sure. That's what this is for.
Yeah. So I think it's important to have resources available for people to digest and then come back and have a conversation about.
Yeah. Yeah. And I'm definitely looking forward to when the mouse study is published. So do we have any idea of the steps that are left to take before you guys, obviously get into clinical trials, but even before you submit to the FDA, like, there's still steps to take. Where are we at in what's left to do there?
Sure. Like Mandy mentioned, or I mentioned too, we've been working on this data package for a number of years. We have most of it done. There's still some finer tuning of a few experiments to do to get all the data that we need. Some more data around, guaranteeing safety and demonstrating that things are working the way we want them to. And then the other piece is we have to also show the FDA that, hey, here's how we're going to make this therapy. So that's a big piece of scaling up and demonstrating that we have a process that envelope and that letter are exactly the same every time we make it.
I'll give the analogy that you gave me that I've repeated multiple times. I love this analogy. BridgeBio is responsible for coming up with the sandwich and deciding which ingredients go into the sandwich, and a company like Catalent is responsible for making sure that 1,000 of those sandwiches are all made exactly the same way every single time. And so what you're talking about is Catalent being able to upscale this in a way that meets you guys' specification.
Exactly. So all of that work is currently going on, but again, we follow the science, we follow the data, so there's no set time period. It's just when it gets completed to the meets.
And that makes sense. Like that makes total sense. I guess my question is, will there be any way for the community to know when that is submitted?
Yes, that's what I was going to add, Ryan. So you beat me to it. We stay in contact with the Galactosemia Foundation regularly, and that would be something that we would communicate in partnership with them and make sure that the broader community is aware.
I would love to know too, so put me on your email list of people you let know, because I'll also let our listeners know, like, hey, this is with the FDA now. Obviously, we won't know the timeline on how long that's going to take to consider, but I think it at least helps as a touch point to know, like, okay, here we go. Like, this is the next part.
Yep.
And that's really helpful. Okay, well, I won't take up any more of your time. Mandy and Clayton, I really, really, really appreciate you guys coming on. I can't say thank you enough. I think, one of the things I was hesitant about, knowing that just pharma has a lot of red tape and stuff like that, is just I didn't want to have you guys on and it be this puff piece for BridgeBio and gene therapy and go BridgeBio, woohoo! I wanted to have a real conversation about it, and I really appreciate you guys being willing to have that conversation. You know, you talked about meeting people where they're at, and I think that goes a long way. And so thank you for being honest with us and telling us some things that we didn't know. And I hope that the community finds this as useful as I found it, and I can't wait to see what happens next with gene therapy and galactosemia. So thank you both for coming.
You're very welcome. Thank you for having us.
Thank you, Ryan.